Hello Year 10. Hopefully you should have in front of you now a copy of the new booklet B1.5, which is on plants and photosynthesis. If you open the booklet to the inside front cover, you can see the syllabus on page 2. It's a very short section of syllabus for this particular unit, and anything that is in dark black, so for example in section B, the end of the paragraph says these are known as limiting factors of photosynthesis. That is the only higher tier content for this particular unit. There are a number of practical tasks that we will be doing next lesson. So there's one in particular where we'll be looking at testing an onion for glucose and testing a leaf for starch. And we'll also be looking at a full investigation into factors affecting photosynthesis. So our little starter activity that we're going to begin with today is on page 3. And it says, starter, what can you conclude? This gentleman up in the top right hand corner, okay, is a Belgian physician called Van Helmont, okay, and he set up an experiment in the 17th century in which he took a willow sapling. A sapling is a very young tree, and he planted this willow sapling in a pot of soil. The willow sapling and the soil, when he planted them together, had a total mass of 90.72 kilograms. He then watered the sapling over the process of the time of about 5 years, and he noticed that the tree gained 74 kilograms, but the soil only lost 0.52 kilograms. So if we look here, we can see the mass of the soil was 90.72. The mass of the soil after 5 years was 90.20. So 90.72 minus 90.20 gives us 0.52 kilograms. Now, what I want you to do is I want you to spend a moment having a think about what can you conclude from this experiment that Van Helmont did. So if you pause the video and write down your ideas underneath. So what is happening over the 5 years? The soil does not provide the plant with any materials for growth. There may very well be nutrients or minerals, okay, in the soil that the plant needs to help it become healthy, to give it nice green glossy leaves and healthy roots and healthy stems, okay, but in actual fact it's not really giving it any food. So where does the willow, the plant, get its food from? Well, hopefully you might have heard of the word photosynthesis, and if we break that word down into its component parts, this first part, photo, means light, and this second part, synthesis, means to make. So if you think of things that you already know that begin with these parts of the word, so photo, photograph, okay, needs light to take a picture, and synthesis, a synthesizer, okay, to make music of a particular style, then that will help you remember what this word means. And in photosynthesis, which we're going to look at today, the plant actually takes light energy and 2 other substances that it needs to be able to make its own food by this process. Turn over the page to page 4, please. So the lesson objectives today are learning to understand what happens in photosynthesis in green plants. This word equation that we can see up on the board, I've put a big kind of like star around it. I'd like you to either highlight this or go over it in a red pen going around the outside, okay, to show how important this word equation is. This comes up on a regular basis in examination questions. It is essential that you know it. And I'm hoping that it should look a little bit familiar, because in the unit B1.2 that we did on respiration and the respiratory system, you saw a very similar equation to this, except it was the other way round. So have a little think just for a minute. What was the word equation for respiration? Pause the video. So I'm hoping on this side of the equation, we should have our reactants, which are glucose and oxygen. And when they react together in cells, they release energy. Remember, we said energy cannot be made. It can only be released, okay? Changed from one form to another. But also, the waste gas that is made is carbon dioxide and water vapour. Now, if we look at that word equation for respiration, what we should notice is that it's very, very similar to the equation for photosynthesis, except all that's happened is it's been switched around. Our reactants for respiration end up becoming our products in photosynthesis, and our reactants in photosynthesis end up being our products of respiration. So if you can remember one equation, either respiration or photosynthesis, you will remember the other one, okay? So at the top of page 4, it says green plants use light energy to make their own food. So this is really important. And they trap that light energy using a special substance that is found inside only plant cells. Chlorophyll is a green pigment and is found inside those cell organelles that we identified in the very first unit, chloroplasts. The green plants obtain the raw materials, in other words, the reactants, okay, that they need to make this food from both the air and the soil. The air should be our carbon dioxide and the soil is going to provide the water. And finally, photosynthesis, as we've learned, can be summarised by the equation below. So make sure you know that equation. So if we visualise what is happening in a green plant, so at the bottom of the page, we've got a picture that looks like this one that's going to come up in a second. So we've already mentioned about light energy being trapped by chlorophyll found in chloroplasts. We've mentioned that it uses this energy to convert the carbon dioxide, there's our CO2, and our water into a sugar that we call glucose. And glucose is a carbohydrate. If we break that word down, we can see carbo is the carbon, hyd is the hydrogen, and any chemical that has these three letters on the end, 8, tells us it contains oxygen. We know that oxygen is released as a byproduct, and that's really useful for animals because we need oxygen for respiration. So in this image, we've got a tree and we've got a little plant on the side here. Here's our source of energy, the sun. It's where all energy ultimately comes from. That is trapped by the chlorophyll that's in the plant, and we can see in both the tree and in our plant there. The carbon dioxide is a gas that comes into the plant through the bottom surface of the leaves, okay, that we can see on this image here. And we're going to see these little structures called stomata or stoma that are little holes, if you like, and pores on the bottom surface of the leaf. The water element gets sucked up through the roots of our tree and of our plant here, and it travels in specialist tubes, and those tubes in the stem are actually called xylem. So my triple science pupils may recognise, okay, this word because they've done some work on leaf and plant structure with Miss Evans. That makes our glucose, which is often stored in the leaves as a different form. The glucose gets converted into something called starch, okay? And finally, the gas, the oxygen, is given out of the bottom surface of the leaf, okay? So this is just a visual summary telling us what the raw materials or reactants are for photosynthesis, how they are taken in from the air for carbon dioxide, from the soil for the water. And it's actually not just one reaction, it's a series of steps, and these reactions are catalyzed, you should recognize that word, speeded up by enzymes. And again, this relates to work that we did right back in that first unit. The energy required to make this reaction work comes from the sun, and chlorophyll is the green pigment which helps to absorb the light from the sun. So plants make glucose first, and then they change that into starch, which is stored. So that's why we will be testing our leaves for starch later on next lesson. So if you look at page 5, you've got a picture of a cross section through a leaf, and you studied this back in year 9. If you look at the diagram, pause the video at this point, have a look at the different layers in the leaf and see if you can answer these questions 1 to 6 for me, please. So if we start from the top of our leaf and work our way down through the different structures, the waxy cuticle on the top of the leaf is there to prevent water loss. So some plants, particularly like the holly tree that is just outside the prep room here, have a thick waxy cuticle to prevent water loss. Question 2, the layer which contains cells with the most chloroplasts is normally in that upper layer just underneath the waxy cuticle, what we call the palisade or mesophyll layer. The upper epidermis layer is transparent. You'll notice there's no chloroplasts in the upper epidermis. This is so that it acts a little bit like a window to allow light through into that top palisade layer for where most photosynthesis occurs. The palisade cells are arranged horizontally. What we mean by that is they're arranged like that, rather than, they're arranged vertically, apologies, like that on the top, rather than horizontally like that. And the reason why is each cell is going to receive an equal amount of light. If they were arranged horizontally, then our cells at the bottom down here would get less light than the cells at the top. Question 5 says, why are there air spaces between the cells in the spongy mesophyll layer? To allow the gases, carbon dioxide, to pass into the cells for photosynthesis. And why can the guard cells change their shape? To close, to keep certain gases in when the plant needs carbon dioxide, or to open to allow excess gases back out again, like oxygen. So we're going to break down, the reactants and the products, and then we're going to be looking at, in our next lesson, as I said, testing the products of photosynthesis in plants. So our main reactants, our raw materials, are water. This is absorbed and sucked up by osmosis in the root hair cells. It gets sucked up the stem in the xylem tissue by a process called transpiration. So the xylem are these kind of tubes that go up the stem. And if you've ever eaten celery, the stringy bits that get stuck in your teeth, they are the xylem tubes, Dissolved in the water, there are normally nutrients or minerals. And higher tier especially, our triple science groups as well as double science need to know that those minerals are not absorbed by osmosis. There should be a key term that you should associate it with this. Osmosis is the movement of water from high to low. Diffusion is the movement of substances from high to low, but minerals move by active transport. And active transport ensures that the substances move from low to high, because minerals are not always in high concentrations in soil. So how does water get into our leaf? If you turn over the page to page 6, this picture, this part down here is very, very similar, to the picture that you've got on page 6. So you've got a slightly clearer picture, which I took from BBC Bitesize. And it says, the water is absorbed and sucked up by osmosis into the root hair cells in the roots of a plant. And this then gets sucked up, the xylem, by the transpiration, or by transpiration. The roots of plants are made up of thousands of tiny root hair cells. And one root hair cell is like this one just here. We've seen these again back in the first unit. They look like a cell that's got an extended part, like a big nose, if you like, okay? That helps to increase the surface area of the root, so it can absorb more water by the process of osmosis and more minerals by the process of active transport. We know that water molecules move from where there are more of them, in other words, a dilute solution, a weak solution, to where there are less of them, a more concentrated solution. And they move into the cell sap that's found inside the vacuole. So this is the vacuole here. So if I was to draw some little water molecules, so I'm going to change to a blue pen, we've got lots of water molecules outside our root hair cell here. So I've drawn 4, 5, 6, 7, 8, make a nice even number, 10 I think we've got there. We might have only 2 or 3 water molecules in our vacuole. So water molecules are going to move through the cell wall and the semi-permeable cell membrane from a high concentration to a low concentration by osmosis, okay? The water molecules will then move again from high to low across each of the cells, moving from cell to cell until they reach the xylem. And in the xylem, because the leaf is constantly losing water by evaporation out the surface of the leaf, so this is up in the leaves, up the top of the plant, it causes water molecules to be sucked, a bit like when you suck on a straw and you suck liquid up through a straw. The water moves up the straw, okay, and into your mouth. So as water moves out of the leaf, that causes more water to be sucked in through this process, okay? So these tubes are called xylem vessels. The reason why plants need support is not just for photosynthesis, but also for support and to keep the cells rigid. So if you remember from the unit on osmosis, when water enters a cell and they swell, they become turgid. So we want plants to remain turgid. We want all the cells in the stem to remain turgid. Otherwise, our stem will become floppy and the plant will wilt, okay? And on a hot, sunny, windy day, the water will evaporate out of these little holes much faster. So you have to make sure that you're constantly watering the plants, okay, to be allow water to be passing up and keeping those cells rigid. So at the bottom of page 6, it says what type of weather will cause more water to evaporate quickly through the stoma. It's exactly the same kind of weather that we hang our washing out in to ensure that our washing dries quickly. A hot day, because heat will cause more evaporation, so the sun being out, okay, provides the heat, and a windy day that's also dry, okay. Windy will take the water vapor away from the leaf, and dry means that there's a bigger difference in concentration gradient, okay. Have a look at page 7. Now I'm hoping Sir will have an example of this so that you can see in front. I've put some celery in a beaker of water, coloured water. All of this is food dye. So this is just red food dye and I think like a greeny blue food dye. And the celery has been left with its leaves on. And you can see that the celery, after it's been left for about 24 to 48 hours, will start to take up the coloured liquid, and the leaves will actually change colour. So they've gone red in this example and a bluey green in that example. If we take a piece of that celery and cut through the celery, this is what you will see inside the celery. I'm going to draw it actually on the previous page. So first of all, the inside of the celery is quite smooth, but the outside layer looks quite bumpy. It's got these kind of ridges. And going up, all the way up, so imagine that this was like a 3D piece of celery. You can see these ridged lines through our celery. And in the middle where it's been cut, you will see a coloured dot. If it's been red food dye, it will appear as a red dot. If it's bluey green, it'll appear as a bluey green dot. So what I would like you to do is I want you to draw that cross section on page 7, okay, next to the picture where it shows some celery being cut. and I want you to add the label to show that these little dots that you can see are the xylem tubes. So pause the video and just draw me a little section of celery, please. So how is water transported in a plant? We've already mentioned the xylem vessels, okay? But next to the xylem vessels, we also have some other tubes. They are called phloem. And the phloem tubes, they carry the sugar, whereas the xylem carry the water. These are held together in one big bundle called a vascular bundle. And the vascular bundles are normally found in the stem of a plant, and they contain two different types of tubes. Xylem, which as we said, carry water. These are strong, thick walls, they're not living, whereas phloem carry sugar and they have thin walls and they are living. So you've got 5 questions. I want you to pause the video and see if you can answer these questions, and then we will go over them. So question 1. What feature of root hair cells enables them to absorb as much water as possible through their roots? They have a large surface area. Question 2. How does water move from the soil into the root cell vacuole? By osmosis. Question 3. Draw a flowchart showing water entering the root hair cell to leaving the stomata in the leaves. Now you've got 4 steps, 4 little spaces to draw this. So I would suggest on the first step that the water passes by osmosis into the root hair cell. That the water then passes from one root hair cell into the one next to it by osmosis until it enters the xylem vessels and it's carried up the stem by transpiration, and then it evaporates into the spaces in the leaf, leaving through the stomata for the 4th one. The structure that water is carried in the stem of the plant is the xylem, and the glucose that is made during photosynthesis also needs to be transported around the plant, and it can actually go up or down. You can see in this arrow here, it can go up to the leaves or down to the roots, okay, where sometimes things like potatoes, root vegetables, carrots, okay, onions will use that glucose to be able to grow, and the structure that carries sugars is called phloem. Turn over the page to page 8. So continuing with our reactants, we've talked about water. We're now going to talk about the gas that's required in photosynthesis. That's carbon dioxide. Carbon dioxide enters the leaves in a plant through tiny holes on the lower surface of the leaf called stomata. So you have got a picture of a cross-section of a leaf. It's got 3, 4, 5, 6, 7 labels that you need to add to it. The labels are clearly shown on this diagram here, so I want you to pause the video and see if you can relate the labels that are in this picture to the labels on your picture on page 8. So if we start on the top surface of the leaf, if you imagine your hand is a leaf and you cut through the hand through the leaf and look at all the different layers, we have our waxy cuticle on the top layer, which we've already mentioned, prevents water loss. We have our transparent upper epidermis that acts like a window into the leaf. There are no chloroplasts here, so light can pass all the way into the main photosynthetic layer. The main photosynthetic layer is our palisade cells that hang vertically down from the upper epidermis so that each cell is getting an equal amount of light. The next layer down below is our spongy mesophyll, which has lots of air spaces between it. There are still quite a lot of chloroplasts down here, but not as many as the palisade, because this is lower down in the leaf. It's going to receive less sunlight. And then finally, our lower epidermis at the bottom here has got little gaps every now and then. The gaps are called the stomata or the stoma as a singular, okay? And the two cells either side of them are called the guard cells, which we're going to look at in a bit more detail later. On your diagram, you have a label with two arrows, one going in and one coming out, okay? I think I've got those the wrong way round, different way to your diagram, so I'll do them the right way round. What might be the gas during the day that's going to pass in, and what is the gas during the day that passes out? So this should be carbon dioxide passing in, and this should be oxygen passing out during the day. At night time though, when there's no light, the plant will not be able to carry out photosynthesis, okay? So instead it will carry out respiration. So we're going to go on and look at these structures that we call stomata on the bottom surface of the leaf. The cells either side, as I mentioned, of these pores are called guard cells, and they can alter their shape. They absorb water by osmosis, okay, and they can swell, and they lose water by osmosis, so they can change their shape. We know that carbon dioxide is required for photosynthesis, so it diffuses into the leaf from the air because there's more carbon dioxide outside the leaf than there is inside during the day. So even though this is a very small percentage, 0.04% CO2 in the air, it will always be higher in the air than inside the leaf. So it causes the carbon dioxide to diffuse in from high to low. So let's have a look at what these stomata look like in a bit more detail. So you've got this image on the top of page 9. We say stomata is the plural, and we've got many of them, and stoma is the singular. They are mainly on the underside of most leaves. However, there are some leaves that have special adaptations that have stomata on both the top and the underside. The way we normally look at these stomata is really simply, we take some regular nail polish and we paint it on both the top and the bottom surface. We allow it to dry, and as it dries, the nail polish takes up the imprint of the cells on the bottom and top surface of the leaf. When we place this underneath a microscope, we can see these kind of structures. So here are the guard cells here. In this example, they're swollen and turgid, and they give us this funny kind of like banana shape, okay, to the guard cells. That opens up the pore. So this stoma is open like during the daytime. Generally at night, or when the plant is not able to get enough water in periods of drought, the guard cells will then become flaccid, they will shrink, and they close together, so the pore is closed. So we see this happening at night, but also when there's not very much water. So if there are any microscopes on the side with pieces of nail polish that you can go and have a quick look at to see these stoma, have a little look. Are they open or closed on your leaves? And write your answer down in the space. And are there more stoma on the top or the bottom surface of the leaf? It should always be that there's more on the bottom surface of the leaf. So it depends if Sir has left some nail polish out that's from top and bottom surface, okay? Can you sketch for me what the guard cells look like in the box on page 9? And pause the video, please. So this is a microscope image if you haven't been able to see any guard cells underneath the microscopes on the side, and they actually look like little eyes, okay? So you can see the stoma really clearly open here, and the two guard cells, if I just annotate over the top, there's one guard cell, there's the other guard cell, okay? And that shows us open stomata. Turn over the page to page 10 for me, please. Now, continuing with our reactants, we also need plants to trap light energy, and we know that they trap this light energy using this green pigment that we call chlorophyll, and chlorophyll is found in the cell organelles, which is our posh word for cell parts, that are known as chloroplasts. And all they do is, there's our carbon dioxide written as a formula, CO2. The light splits the carbon from the oxygen. Our other reactant, water, the light splits the hydrogen from the water, and it's going to recombine these particular elements, carbon, hydrogen and oxygen, to make our glucose, our carbohydrate, with this particular formula, and it also releases oxygen. Now currently that word equation or formula equation is not balanced. To balance this equation, there are 6 carbons, so we put a 6 in front of the carbon dioxide on the reactants. There are 12 hydrogens, so 6 times 2 will give us 12 hydrogens. So we've managed to balance the carbon and the hydrogen, but our oxygen, we've got 6 times 2 is 12, plus another 6 is 18, and yet on this side we've only got 8, 6 plus 2. So all we do is put a 6 in front of the oxygen. 6 times 2 is 12, plus 6, okay, is going to give us 18 again. These are what the chloroplasts look like, and you can see the lovely green colour inside them, the chlorophyll that's going to trap the light energy. So to summarise, to finish this lesson on page 10, you have got this closed procedure, okay, to be able to use these words that are listed here to fill in the gaps. Pause the video to add those in. So during photosynthesis, light energy is absorbed by chlorophyll, a substance found in chloroplasts. This energy is then used to convert carbon dioxide from the air and water from the soil into a simple sugar called glucose. Oxygen is also produced and released as a gas. So check you've got those in the right spaces and give yourself a mark out of 1, 2, 3, 4, 5, 6, 7, 8 out of 8. We're then going to answer these next 3 questions. Question 1 says, work out the path taken by a carbon atom as it moves from being part of the carbon dioxide in the air to being part of the glucose and then a starch molecule in a plant, and write the route in the correct sequence below. Explain, which means state why carbon dioxide diffuses into the leaf. Think about it, diffuses, okay, think about the concentration gradient. And at night, the stomata usually close as carbon dioxide does not need to diffuse into the leaf. Why is this? Pause the video to attempt those questions. So for question 1, if the carbon dioxide is in the air, it's going to travel through the stomata by diffusion from high concentration to low concentration into, okay, the spongy mesophyll part of the cells, where it's going to travel into the palisade cells to be used in the process of photosynthesis to make glucose, which is then going to get stored as starch. Explain why carbon dioxide diffuses into the leaf during the daytime, because it moves from high to low concentration gradient. So there's more carbon dioxide during the day outside the leaf, and it moves inside the leaf to where it's lower. And at night, the stoma close, and carbon dioxide does not need to diffuse into the leaf because there is no photosynthesis. Instead, there is respiration occurring in the leaf. So next lesson, we will be looking at 2 practicals. We'll be looking at testing an onion bulb for glucose and testing a leaf for starch. Thank you, Year 10.